So Ruthie, prior to Medidata, you were at the FDA for 22 years. Could you provide your perspective on regulatory submission risks that arise from poor data quality?
Yes, thank you. I'd love to do that. And honestly, I've seen a range of possible outcomes based on data quality. The most common, and of course the one that everyone is aiming for, is that the FDA finds the data quality at least acceptable, and data quality issues do not impact the review of the marketing application. However, in the cases where data quality is questioned, the agency often requests additional efficacy or safety sensitivity analyses that exclude problematic patients or sites, and the consistency between those analyses and the overall original analyses will be important. Often, though, the lack of power associated with the reduction in the sample size is very problematic. But even if the consistency is achieved, the data quality issues can be costly in that they compromise the reviewer's opinion of the application and just invite additional scrutiny of the application in other areas. In rare cases, the identification of a small number of severely compromised sites could bring the entire study into question. I have seen cases where the agency concludes that problems uncovered at certain sites are likely to be systemic, and so that the entire study is suspect and not reliable. Very serious data quality issues can sometimes disqualify an application from presentation at advisory committee meeting, and the rationale there is that the experts on the committee should not be expected or could not be expected to resolve data quality issues, and they should not be asked to opine on data that is faulty. And then in the most severe cases, the agency may reject the study, request confirmation of efficacy and safety with an additional trial, or simply not approve the product. So there's some very serious outcomes with problematic data quality. And then on the next slide, what I'm showing you is one example of how FDA handled an important phase 4 study with data quality issues. This example was made public by an advisory committee that was held last fall. The study in question here was a large phase 4 safety study required by FDA as a post-marketing commitment to examine the occurrence of neuropsychiatric events with the use of the product. And I'm not sure how many of you are familiar with the setup of an advisory committee, but it's essentially a day-long meeting focused on the single application, and the day ends with a vote about the critical aspects of the discussion. So after this day-long discussion, much of it which was centered on data quality issues, the question posed to the committee of experts by the FDA about data quality, the committee had these responses. They commented on the possible underreporting of adverse events, and they had heightened concern because of the unexpectedly low adverse event rate in the placebo group. Some commented that excluding problematic sites from the analysis doesn't get at the fundamental issue of underreporting adverse events. And in addition, many of the members of the committee suggested that their faith in the study had been compromised, and I think we can't underestimate the impact of something like that. And so just noting, while most of the analyses of the observed data were largely positive and indicated that the product did not increase the risk of neuropsychiatric events, the advisory committee vote was split with 10 in favor and 9 against removal of the black box warning for this product. And the rationale for the negative votes was very often concerns around data quality and what conclusions might have been reached if complete and accurate data were available. So this is one example where data quality concerns played a very important role in the regulatory outcome of the submission.
Stacy, I want to turn to you now to talk about the solution that Medidata has, which uses a novel approach to help the industry comply with ICH E6R2. Could you tell us about that solution?
Sure. On that next slide, Kalyani, yeah. As Kyle talked about in the poll, one of the pieces that people are struggling with is identifying the right technology, right? And you can also remember he spoke about one of the ICH new requirements to use statistical analysis or analytic approach to the identification of data trends within and across sites. And that's something that is really core to the central piece around centralized statistical analytics. As we've mentioned a couple of times, it's a technology based on machine learning. And it leverages machine learning to take in all of the data in the trial. And that data in the trial is, the CSA is source agnostic. So you can bring in RAVE, EDC, electronic data capture data. You can bring in other forms of EDC. We also integrate and bring in the laboratory data, ECG data, a lot of types of eDiary data, ePro, and other patient-centric sources. We bring those sources into CSA and integrate them. That the advanced algorithms and the machine learning then hit against all of the data in the study, and it essentially compares every variable to every other variable in the study to learn what normal looks like for that trial. And then anything that doesn't fit within those patterns that have been created on the study gets flagged as a potential anomaly to be investigated. And you can see here a graphic of one of the visualizations that's common in CSA, where on the y-axis you see the study day of resection, and this is a solid tumor oncology study. So the study day of removing that tumor. And then on the x-axis, you see the study day of diagnosis. Those 2 points that you see the arrows beside, those are examples in which CSA is flagging that there are 2 outliers there in which patients are being diagnosed before their tumor is being resected, which is not possible, right? You have to have the solid tumor removed and tested before you can make a diagnosis on it. And so this is an example where CSA clearly sees a relationship between study day of resection, study day of diagnosis, without you as the end user having to tell it to look for it. And it sees a problem with a couple of patients where they're being diagnosed in a manner that's just not logical or accurate. A lot of times what we're seeing in this situation is that electronic edit checks have already been built around these and fired appropriately at the site, but the sites are becoming so inundated with edit checks that many times they're responding in these cases to say that the source data is correct, and then the CRAs, the data managers, are also inundated with the responses. And so we're seeing them just blindly accept those responses, closing out the queries, and then these data errors are remaining in the system. Again, these are examples that regulatory agencies would probably look at if they saw a pattern in this. So as you see on the left-hand side, as I said earlier, CSA is source agnostic, so you can bring in multiple sources into the system. It detects and looks for patterns, and then we're also building in that critical workflow management that Kyle mentioned earlier to ensure that our cross-functional teams are able to work together in a robust manner within the system. So it's been used across large and small pharma. It's been used in some of our partner or CROs, and so this is something that we see is going to really start advancing our industry the way it has other industries.
So turning to Ruthie and to further talk about CSA and how CSA could have addressed some of these issues, Ruthie, can you describe for us, in the example that you discussed earlier, how CSA could have addressed some of these issues?
Sure. Some of you may be aware of an advisory committee that was held last fall to discuss the data quality of a large phase 4 safety study. This study was a post-marketing requirement by the agency to examine the occurrence of neuropsychiatric events with the use of the product. And if you've ever seen an advisory committee, you know it's a very lengthy discussion, often at least a day or more. And in this case, after that discussion centered on data quality issues, the day ended with a direct question posed to the committee of experts by the FDA regarding the data quality. And the experts on the committee commented on possible underreporting of adverse events, and they had heightened concerns because of the unexpectedly low adverse event rate in the placebo group. Some members commented that excluding the problematic sites from the analysis really wasn't sufficient because that wouldn't get at the fundamental issue of underreporting of events. And in addition, many members of the committee suggested that their faith in the study had been compromised by the various data collection errors. And while most analyses of the observed data were largely positive, indicated that there was no association with use of the product with neuropsychiatric events, the advisory committee vote was split, 10 in favor and 9 against removal of the black box warning from the product labeling. And the rationale for the negative votes was very often concerns around data quality and what conclusions might have been reached if complete and accurate data were available. So as was mentioned in the pool in this webinar as well, the reporting of adverse events is a key issue. And so what we're showing you on this slide is an example of how CSA can identify possible underreporting of adverse events. So this is a real but anonymized case study. And in this case, CSA identified that certain sites in Italy, which made up more than 10% of the study population, were significantly underreporting adverse events. On the left of this slide, we see a display of the sites ordered by their adverse event rate per patient year, and 3 sites are identified with possible underreporting of adverse events. From the display at the top of the slide, we see that these sites reported no headaches. That's the red square in that display. And this is unusual in that headache was the most commonly reported adverse event in the overall study at a rate of nearly 20%, which is shown with the green triangle. The display on the bottom of the slide illustrates the identification of several patients with very low hemoglobin relative to other patients in the study, and yet no anemia event was reported for these patients. And most of these patients were enrolled at the 3 Italian sites of concern. So another concern at that advisory committee that I mentioned was the possibility of a misunderstanding of the data collection tool. And if we go to the next slide, we'll show you a case where we identify a similar error with CSA. So the error that was mentioned at the advisory committee was a citing of results of pairs of variables that seemed inconsistent. And indeed, this is a very common finding for us as well, especially with reporting of quality of life measurements by the physician or patient, for example. We have many examples where components of the measure that should be positively correlated, in certain cases they appear to be negatively correlated. And we see these kinds of errors very often. And even, we sometimes see them even in more objective measures, such as what's shown on this slide. So here CSA detected the expected relationship between ECG heart rate and respiratory rate, but highlighted a site on the right of this slide where that relationship was an exception. So it was essentially the reverse of what was expected. So in this case, this allowed retraining and instrument checks at that site. And on the next slide, I want to share with you another example similar to this. This slide shows patients with no variability in a clinician-reported outcome, the Global Clinical Impression Score. And these measures were taken across numerous study visits, and so it seems unlikely that a measure would not change at all over the entire course of the study. This could represent sloppiness in the study conduct, or it could also, as was discussed at the advisory committee, represent a simple misunderstanding of how the data reporting is supposed to be conducted and the data reporting mechanism. In this case, CSA also highlighted that most of these subjects with 0 variability were from a single site, suggesting that retraining of that site may be warranted. And then on the next slide, as a final example of errors that may be lurking in the data you're working on, we will show you a case identified by CSA that strongly suggests data fabrication at a single site. This is, again, a real but anonymized illustration of the case. So at the top of this slide, we have something called the patient similarity plot. And here we're using this plot to alert us to patients that appear to be overly similar in the data they're reporting. And the proportion of overly similar patients at each site are shown in this histogram, and they are highlighted in red. There's one site that we have circled that has 5 patients where all 5 patients appear to be overly similar. And we can also see that the follow-up time for all 5 subjects are very similar, differing only by a few days from one patient to the next. Moving sort of down and to the left on the slide, And further investigation revealed that certain lab values were unrealistically similar across patients and time. And in the top display there, you see the 5 patients' lab values overlaid in a single plot, and they are nearly identical, as opposed to on the bottom, a site with 5 patients with more typical between patient variability. And then moving to the right, we can see that these 5 patients had nearly identical spacing of their visits across the entire course of the study. And this was unusual compared to the rest of the patients in the study, where there was some natural variation in when patients could be brought in. And then finally, I'll point out an unusual result in the AE frequency. There was a rare event that occurred in only 2% of the study subjects overall, and yet it was reported to have occurred in all 5 patients at that single site. So in these ways, CSA can provide a comprehensive look at data quality, allow early identification of possible problems, and correction and retraining of the site as needed, so that errors and criticisms of the study results based on data quality, such as were discussed at last fall's advisory committee, are avoided.